Okay, thank you. Thank you very much. I hope you can hear me at the back. I just want to spend about 5 minutes or so with some concluding remarks from the perspective of a regulator, from the perspective of the European Chemicals Agency. In my role, really, as co-chair, I was co-chair with Ian for part of the time for the scientific expert panel of Surat 1. And then I'm going to finish with my last slide with a few personal remarks from this very interesting project that I've been involved with over the last 5 years. Time goes very quickly. So to begin by explaining what the Chemicals Agency does. We're the European Chemicals Agency in Helsinki, and what we do is we regulate industrial and household chemicals and biocides. For chemicals, that means the REACH system, the REACH registration and evaluation scheme, which includes risk management activities as well, so there's the two parts to that. Then there's the classification scheme, which is hazard communication to workers and the public about the immediate hazardous properties of chemicals and packaging and labelling. And then there's the biocidal products regulation, which are products that are intended to be biologically active, such as fly sprays, slimicides, rodenticides, that sort of thing. And if you think about when you're using information on the properties of chemicals, they're used in two contexts for these regulations, this stuff that we do with chemicals and biocides. The first way is to do a definitive assessment of the individual properties of an individual substance. So that could be for classification, hazard identification, hazard quantification, and risk assessment of the individual substance. And for that, you can't have a big error rate. You want pretty good quality information with very few mistakes, basically. The second purpose, which is especially important for REACH, is to do for priority setting, screening and priority setting. So you have a universe of chemicals and you want to pick out those that you want to do something about that require regulatory intervention. So I'm thinking about using new approach information, new non-animal data in those two contexts. And that, of course, is reflected in a lot of other regulatory schemes throughout the world, US EPA, etc., and our sister agencies in Australia, Canada, and so on. So if I now focus on definitive hazard assessment for an individual substance, that's the first one I've been talking about, the first main purpose, ECHA strongly encourages registrants to comply with their legal obligation to not do any new animal testing unless it's absolutely necessary. New animal testing should only be done as a last resort. And they can look at our guidance on the issue and our advisory documents. We help with the OECD toolbox. We've got training, so look at our website. So it's really important. We take it really seriously to remind companies of their obligation not to do any new animal testing unless it's absolutely necessary. But if a company is going to use non-standard methods, basically the point is that prediction, the predicted property, must be good enough for risk assessments and classification, which means, broadly speaking, that you're providing equivalent information to the standard classical toxicology test that you're omitting, that you're substituting. And we found in practice, and again this is common sense really, knowing the state of the art in regulatory science, the absolute best way to do this at the moment for complex endpoints, such as the 90-day repeat dose oral toxicity study, is read across. That means you have several substances of similar chemical structure. The scientific justification is they're likely to have similar biological properties. Therefore, you can read across from one to another, so long as you've got some sort of supporting evidence that the biology really hasn't changed by shifting the chemical structure in a subtle way. The other technique, that's the regulatory technique that's used moderately often, is weight of evidence, which is in many ways a sort of extension in some cases of read across categories and read across. So that's the background, and then we say, well, why should ECHA be involved at all? Why should I be on the scientific expert panel? I was invited just at the beginning of my time at ECHA, actually. I've been there 7 years now, and just at the very beginning I was invited to join the expert panel. I thought, oh, well, and I got agreement from my boss, the executive director. So why did he agree? Well, the basic point is ECHA has a regulatory science strategy, and one of the motivations is to serve as a hub of scientific excellence in regulatory science. And that basically involves in active participation in the regulatory science community. And you're the science community, if not necessarily purely the regulatory science community, but we want to bring a bit of regulatory reality and interaction with you. And one of the big things is to have a clear concept of how we see R&D is needed, often more the method development side, but fundamental research as well. So we have a clear problem formulation. And one of the things that we're very keen on is combination of evidence in a biologically rational way. And the third thing we do is our topical scientific workshops every year. And I'll tell you about the one next, in a minute, I'll tell you about the one we're doing next year that's taken inspiration from the work of Surat. So, that's the theory about why I engaged, why ECHA engaged with Surat. And what did we actually do? Well, I think I can pat myself on the back a little bit. We can pat ourselves on the back at ECHA and say, yes, we did input to Surat to articulate the regulatory application of the work you've been doing, that Surat's been doing. And we came across, we were instrumental in saying that you can actually have an immediate application of new methods to support categories and read across, to enhance categories and read across, and that can actually begin now. And of course, we're very interested in the next registration deadline, 2018. We want to influence registrants' behaviour now, not in 10 years' time. And so one of the practical outcomes is we're having next year's topical scientific workshop is on new approach methodologies next April. And the first day is concentrating on using new approach methods to enhance categories and read across. The second day thinks about prioritization and then general approaches. And another almost unforeseen benefit, a serendipitous benefit, was the enhanced cooperation that we have with the Joint Research Centre and EFSA, because we have memorandums of cooperation with these organisations, and it's really, really important in our engagement with the academic and regulatory science community that we engage with these key actors. And Surat has been a focus of that work, so it's been very, very successful in that way as well. And we're continuing, the final thing is we're continuing to interact with the regulatory science community by supporting the new follow-on project, EU Tox Risk. So now that finishes the sort of official part, and these are my very, very informal personal reflections as 5 years as a SET member and a co-chair for part of that time. I must admit, originally, I was a bit sceptical or uncertain. I put uncertain on the slide, but I meant a bit sceptical about what actual impact we'd get from a regulatory perspective, practical application for industry and for ECHA, what we'd get out of this thing, and what actual impact I'd be able to have personally as a member of the SEP. Well, that second part, I think I've already just mentioned, because I think we did manage to influence the work, and I think it's been very productive. And it actually emerged that there was brilliantly excellent cooperation between the different parties, the researchers, industry, regulators, and internationally. The international context has been fantastic, really, really important. And now, the next bullet point is, this is, in many ways, I think that Surat has been at the right place at the right time to do this, to have a good achievement, for the reasons I'm explaining briefly in the bullet point. But in actual fact, nobody's actually said why it was called Surat, and it's basically named after the painter. And the painter, of course, Seurat, the English say Surat, put the T at the end because we can't pronounce French names, made little dots and came up with a picture that meant something from little dots, from little pieces of evidence. And I think that in many ways this is exactly what happened. We were at the right time to do this, because if you're going to have a picture, you've got to have the outline. And the outline in terms of Surat work is a better understanding of biology, much better understanding of the underlying biology of how chemicals actually cause problems, toxicity in human health. The second element is the little dots that make up the picture, and that's these lots and lots of new tools and techniques that provide a huge amount of information, either high throughput screening or vast amounts of data from high information content, omics type stuff. And then the third bit is is, if you imagine the painter actually putting them on the page, but putting the dots in the frame, and that's the ways of describing that evidence within the biological framework. And there are regulatory techniques that have fairly recently developed. There's a knowledge technique, basically, of combining evidence called adverse outcome pathways, or mode of action, mechanism of action. And that was at the right time. This was just there at the right time to be clinched by the work that Surat did. And the regulatory equivalent of that is this IATA, the Integrated Approach to Testing and Assessment. And overall, this has led to the Surat conceptual framework, which in actual fact is a sort of very general version, a generic version of an IATA, because those have only really been applied in very limited cases for simple endpoints where there aren't lots of different complicating factors. And the conceptual framework manages to bring together a way of structuring evidence, the little dots, to give an overall picture. So the other point which I reiterate is I was very pleased that we're actually having a direct and immediate regulatory application, practical application from the work of Surat, the categories and read-across enhancement stuff, and the TTC stuff that can actually be actually enhanced and be used by the cosmetics industry. I think that's really, really good. So I conclude by saying it's been a great privilege, actually, a great pleasure and a privilege to be working with such fantastic colleagues within the Surat network and on the scientific expert panel. It's been fascinating science work, and I've really, really enjoyed being involved in this. And I hope to see some of you, if not all of you, at our April Topical Scientific Workshop on New Approach methods next April. So thank you.